Hey, let's talk about bacterial gene regulation. So now we're looking at how bacteria can regulate different gene production. We don't need genes to continuously be expressed, especially if we're not using those genes. And so we're always turning on and off different genes. In general, bacterial genes are found in operons, where you have multiple genes coded for that work together. So an operon is a set of genes that includes an operator region, a promoter region, and then the structural genes themselves. Hello. Maybe. There we go. So there are two major regions in an operon. There's a control region and then a structural region. The control region controls transcription, whereas the operator region, whereas the structural region is the region that codes for the genes. So here we have that control region where we have a promoter site and an operator site. Promoter site is where RNA polymerase will bind. Oh, and I have it there. The operator site is where genes that control the running of the operon bind. And then we have the structural genes here that actually function together in controlling whatever, producing some response. So there are three types of genes that we typically think of. Constitutive genes are genes that we're always producing. So these are enzymes that would be working in metabolic pathways like aerobic respiration or the glycolytic pathway. Then we have inducible genes. These are genes that are typically not produced unless we need to produce them. And so they're turned off until we need to turn them on. Repressible genes then are genes that are typically produced until we don't need them anymore. So they're typically turned on until we turn them off. So inducible enzymes are found mainly in catabolic pathways. And so they're only turned on when we have something we need to break down. Whereas repressible enzymes are found in anabolic pathways. So if the pathway if we don't need some enzyme or some material for the anabolic pathway, we've got enough there, we turn it off for that reason. And so we have proteins that are called repressor proteins that stop transcription from occurring. We also have activator proteins that can enhance transcription. So when we talk about repressor proteins, these are used in what we call negative regulation, whereas activators are found in positive regulation. So negative regulation is how we stop protein synthesis using different types of repressor proteins. So there's induction and repression. Both are negative regulation forms, and they both use repressor proteins. In induction, a repressor protein is bound and blocks transcription, and the only way it comes unbound is if an inducer binds to the repressor. Whereas in repression, a repressor binds to the operator site only if a co-repressor binds to it. And so let's look at what that looks like. So this top one is showing induction. So here we have a repressor protein bound to that operator site. RNA polymerase binds, it can't produce the protein. It can't make that structural gene. because the repressor is stopping it. But if an inducer binds to the repressor protein no longer can bind to the operator site, and now RNA polymerase can produce that structural genes. In repression, the repressor protein cannot bind to the operator site normally, and so RNA polymerase can bind at any time and produce a copy of those genes. But if we have a co-repressor that binds to the repressor, now the repressor changes shape and it can now bind. So up here, think of the inducer as maybe something we want to break down. And this is a catabolic pathway. And we're going to produce genes that break down that whatever molecule this is. Down here, think of this as one of the products of some anabolic pathway, and we have a lot of it, and so it binds to stop producing more of itself. So now let's look at how activators control gene expression. Activators are also known as your positive regulators, and what they do is they enhance the ability of RNA polymerase to bind to that promoter site. In many cases, RNA polymerase can only bind if an activator binds to the activator binding site. And so if you don't have an activator bound, then the RNA polymerase can't bind anyway. So it doesn't matter if there's anything on the control region. Hold on. Hello. So now let's look at an operon and how it has both positive and negative regulation. It uses both repressors as well as repressor proteins as well as activators to control gene expression. The lac operon is one of the most well-known operons. So near the operon is a region that controls the production of the repressor protein. And so this is known as the I gene. And the I gene produces repressor proteins. And then when no lactose is available, the repressor protein will bind to the operator site. And so if the operator site is bound, it doesn't matter if the RNA polymerase binds to the promoter region, because there's no ability for that gene to be produced because the repressor is on there. So that is a repressible gene. So it's being repressed, or not impressible gene. It is being controlled by a repressor protein. That's what I want to say. So that is negative regulation. It is an inducible operon, which means if an inducer comes along and binds to the repressor protein, then the repressor protein will move off of the lac operon. So here we have the lac operon model. We have a promoter site. This is where RNA polymerase binds. The operator site, this is where the repressor protein sits. And then this is the structural gene region, okay? RNA polymerase, even if it binds to the promoter region, if the repressor protein is in place, you can't produce the structural genes. The only way the repressor or the genes can be produced is if there is an inducer that binds to the repressor, changing the shape of the repressor protein. So there you go. Then when RNA polymerase binds to that to the promoter region, it can actually run and produce the structural genes. But the operon also has a cap binding site. This is a site where the activator binds. So there's an activator site right next to the promoter site. If the activator binding site is not bound by the cap protein, then the RNA polymerase can't bind either. So this is a positive regulator. So cap protein is modulated by the amount of cyclic AMP. So cyclic AMP is inversely correlated to the amount of glucose. So cyclic AMP will bind to cap if it's available, but if cyclic AMP is not available to bind to cap, then cap can't bind to that promoter or to that activator region. So if you have a lot of glucose in the environment, that means you have very little cyclic AMP. So let's just put increase glucose equals a decrease in cyclic AMP, okay? Because they're inversely correlated. That also means that if you decrease glucose, then you're going to increase cyclic AMP. Make sense? And so here is that cap binding site. There's the promoter site, right? So here, if cap has cyclic AMP available, then you're going to have cap bind to that cap binding site, and then RNA polymerase can bind to the promoter site. But if, and I'm just going to come down here, if cap doesn't have cyclic AMP bound, it can't bind to the promoter site, and then RNA polymerase can't bind to the promoter site. So cap can't bind to the cap binding site, the activator site. Therefore, RNA polymerase can't bind to the promoter site. So let's look at the 4 different type or mechanisms that control this lac operon. If you have lactose, but you have no glucose. Lactose is the inducer. So if lactose binds to the repressor protein, it changes the shape of the repressor protein. The repressor protein won't be able to bind to the operator site. In that case, the operator site is opened. But you also have no glucose. So remember, cyclic AMP is inversely correlated to glucose. So glucose and cyclic AMP, right? So if you have a lot of glucose, you have little cyclic AMP. If you have little glucose, you have a lot of cyclic AMP. So if you have no glucose and you have a lot of cyclic AMP binds to the cap protein, which then allows the cap protein to bind to the cap site. This allows the RNA polymerase to bind, and you can produce that transcript. Now look at here with no lactose and no glucose. So again, no glucose means you have a lot of cyclic AMP. Cyclic AMP binds to the cap protein, so cap binds to the cap binding site. But there's no lactose, so you don't have that inducer, which means that repressor protein is going to bind to the operator site. So you're not going to produce the transcript. Here you have lactose and glucose in the environment. So there's lactose, which means lactose binds to the repressor protein, and the repressor protein comes off of the operator site. But there's also glucose, so you have a lot of glucose, which means you don't have cyclic AMP, so cyclic AMP doesn't bind to the cap binding site, so there's nothing bound here. So RNA polymerase can't bind and produce its transcript. Now you have glucose and no lactose. So we have glucose, which means we have no cyclic AMP, which means cap can't bind, okay? We also have no lactose, which means there's no inducer binding. So the repressor is going to bind to the operator site. So again, we're not going to produce the structural genes. So the only time you produce the structural genes in the lac operon is if lactose is present and there's no glucose. Then you will actually allow the lac operon to run, and you can produce the proteins needed to break down lactose. That is the end of my lecture, so I'm going to stop here. This was a longer video, but covered the whole lac operon. So I will talk to you later in our next chapter. Bye.